Okay, sensation and perception, chapter 4. We'll start off by making a distinction between sensation and perception. We'll operationally define our terms so we know what we're talking about. Sensation, the stimulation of sense organs by the outside world. Perception, the selection, organization, and interpretation of sensory information or sensory input. So what we're talking about here is this. Sensation is the actual gathering of raw data by your eyes, your ears, the pressure and temperature receptors in your skin, etc. Perception is the processing of that information by the cortex. So what this means is that in a very real way, although your brain needs the data provided by your eyes and your ears and so on and so forth, the actual experience of vision and sound is a brain-based phenomena, not a sensory organ-based phenomenon. Put slightly a different way, you see with your brain, you hear with your brain, you don't see with your eyes or hear with your ears. That's just those organs, the eyes and the ears are just supplying the data to the brain where the actual experience of vision and hearing takes place. A good example of how this is the case is something I may have referred to earlier in the semester when we were talking, actually last class period perhaps, that we can actually create the experience of vision by directly stimulating parts of the visual cortex, the part of the cortex where vision is created. In the back of you, touching the back of my head because this is where the visual cortex is. During brain surgery, say to remove a tumor from the visual cortex, they may very well keep the patient awake and use electricity to stimulate the brain, or use electricity to make a distinction between, are we cutting the tumor, or are we getting ready to excise a tumor, or are we getting ready to damage part of the brain? And if you're stimulating the tumor, the person has no experience. As you excise that tumor and remove the cancer, and you begin stimulating things that are no longer tumor and are actually part of the visual cortex, part of the brain itself, the person will have seemingly spontaneous visual experience. You can cause them to, quite literally, they'll keep the person awake, and the neurosurgeon will be talking to the person to get feedback from, are you having an experience here? And the person, when the brain rather than the tumor is stimulated, will give the surgeon the feedback that, oh wow, yeah, here, I see my mother's face, or look at the puppy, or whatever. And they're having a very real perceptual experience, even though their sense organ, in this case the eyes, is not being stimulated. So we see with our brain rather than our senses. Our senses, eyes, ears, the skin, the tongue, is simply providing the data to our brain for processing. In a similarly counterintuitive event, we experience the world. The slide there says we do not see the world as it is, but rather through receptive fields. What the slide really should read is, we experience the world not as it is, but through our receptive fields. For example, you can see here on this depiction of the spectrum of electromagnetic radiation that we call light, that the portion of the spectrum that we make use of for vision is actually a minuscule segment. You can see the rainbow a visible light there, and if you follow the graph upward, you can see that it's actually a very narrow band of the whole spectrum kind of crammed in between ultraviolet and infrared wavelengths. We make use of a very narrow band of wavelengths, which basically means that when you have open eyes and light is reflecting off of things and entering the eye through your pupils and stimulating your retina, all of which we're going to talk about in a few minutes, you are not making use of the vast preponderance of the electromagnetic radiation that is light that is entering your eyes. You're just using a little narrow band of it. So there's all this information about the world, hypothetical visual information about the world that your eye is simply not designed to absorb and use. Similarly, our ears. You can see here the what's the word? Not depiction. The distinction between our fairly narrow auditory range and the auditory ranges of other animals, dogs, cats, dolphins, bats, etc. The same sound waves are stimulating the eardrums, are impinging upon the eardrum of an elephant or a dolphin, as are stimulating our eardrums. But the neurological mechanism that is the inner ear, the place where we actually pick up the data and translate the data of sound into something that our brains can use to experience hearing, we're making use of a much narrower band of wavelengths. The dolphin, the dog, hears significantly more information than we do. Our ears are simply not designed to pick up all of the information that is available to other species. Again, what this means is that we are not hearing the world as it is. We're hearing the world as our processors, our information gathering tools allow us to hear it. And we're hearing the world as our ear is capable of picking up, detecting data, detecting sound wave differences, and then being processed by the brain. So we experience the world not as it is, but as our senses allow us to experience it. We do so through a fairly limited number of types of neurons. There are basically 4 general categories of sensory neurons that comprise our receptors. Photoreceptors, which are the neurons in your retina in your eye that are sensitive to light energy. Mechanoreceptors, which are the neurons in your skin, in your inner ear, that are stimulated mechanically, that they actually are moved, they detect pressure, they detect movement, and we'll talk about the exact mechanisms for that here in just a little bit. Chemoreceptors, which are stimulated by chemicals. These are the neurons that comprise your taste buds and your olfactory bulb, the neurons by which you smell. And then thermoreceptors, neurons that are sensitive to temperature. So these 4 types of neurons, photoreceptors, chemoreceptors, mechanoreceptors, and thermoreceptors, take the raw data of the world and turn light, sound waves, pressure, temperature, and turn it into an on-off signal. action potentials, APs, action potentials. And these action potentials are sent along the afferent neurons, the neurons that are part of the sensory system, to the parts of the sensory cortex for processing. So all of the information that is being delivered to the various parts of the cortex where the information is processed, all of that information, all that sensory information is broken down into 4 basic characteristics. What mode is the information? And basically what this means is that information that is processed as vision is the information that is coming from the eye, that is coming along the optic nerve. Information that's coming along the auditory nerve from the ear is processed as auditory information. And interestingly, to give you an example, you rub your eyes and you press on your eyes, which everybody tells you is not good for you. But you press on your eyes, and if you press hard enough, what do you get? You get flashes of light. You have a visual experience to pressure on your eyes. You experience what is actually not light, instead is pressure as you're pushing on your eyes, but you experience it visually because that's where the information is coming from. The neurons are being, the pressure on the neurons is being interpreted as the experience of light. So modality, where the data is coming from, determines what mode it is experienced as. Is it experienced as sound, touch, hearing, vision, etc. It also is broken down into the characteristic of intensity. How bright is the light? How loud is the sound? How heavy is the pressure? And this is essentially communicated by the rate of firing. The brighter the light, or the more intense the hue, or the purer the sound, or the louder the sound, is basically a function of particular neurons being stimulated to greater or lesser degrees, and the way that's communicated is simply how quickly they're firing. A brighter light is experienced by certain neurons firing more frequently. A louder sound by certain neurons in the auditory pathway firing more frequently or less frequently. The third of the characteristics that sensory information is broken down into is location. Where is the information coming from? And this would be something like in your visual field, what parts of the retina are being stimulated. In the inner ear and the auditory nerves, what segment of nerves is being stimulated? Is it nerves that are particularly sensitive to higher pitched sounds or lower pitched sounds? So what location are they coming from? Quite literally, in terms of where in the inner ear, where on the retina, where on the skin, these kinds of things. And then the third of the characteristics is duration. How long is it lasting? Is it very brief, or is it longer? So all of the information that is processed by your brain and turned into a very, very rich perceptual experience of vision and hearing and touch and taste and smell, and all of it you can break down into these 4 basic characteristics: mode, intensity, location, duration. And I'm not going to do much with this right now, but I wanted to give you a sense of what I was talking about when I was referring earlier to the sensory cortex. I wanted you to have a basic picture in your head of where the visual cortex is auditory cortex, olfactory, which is the cortex where your sense of smell is processed, gustatory, taste, somatosensory, which is the tactile information, and motor will eventually become motor information when it's sent to the motor cortex, and the vestibular cortex, which we're going to talk about in just a second. So, corresponding to the areas of the cortex, we have 5 sensory systems. You'll see up there on the slide that I have that I write it as 5 plus 2, and I don't mean that literally. I'm wanting to make the point that the system that we associate with the experience of touch is a little bit more complicated than we generally acknowledge. So our 5 senses, the visual system, the auditory system, the olfactory or the smell system, the gustatory or taste system, and then what we talk often about it as the touch system, but it's really better to talk about it as the somatosensory system, the body sense system. And we'll start off by talking about the somatosensory system and why that is a little bit more complicated than it initially appears to be. So the plus 2 that comprise the somatosensory or are part of the somatosensory system. So bear in mind, you have the touch system, right? You have the pressure, the mechanoreceptors that are pressure receptors in your skin. You have the thermoreceptors that are temperature receptors in your skin. This is how you're given feedback about something touching you or you touching something, say with your fingers, and given information about how hot something is that's being touched. We think about that as our touch system, and it is, but the reason I'm saying that the somatosensory system might be a better term to describe it is because that tactile system includes 2 additional inputs. The first one that we'll talk about is what we call the kinesthetic system, which gives us the experience of what we call proprioception. And proprioception is your experience of body position and strength of movement. This is the sensory information that allows you, if you get pulled over for driving like a lunatic and at 10 o'clock on a Friday night and the cop gets you out and they make you do the drunk test, right? Well, that drunk test, at least the way I've always seen it depicted, is you walk the line, right? And you walking the line is, we're going to talk about that sensory system here in just a second, the sense of balance. But some of the other things that they have you do are things like, they'll have you put your arms out and close your eyes and touch your nose, right? With your fingers with your eyes closed. And we don't tend to think about how remarkable this is, but it actually is fairly remarkable that you have a sensory system that gives you information about where exactly in space is the tip of your finger and where exactly in space is the tip of your nose, and you can connect the 2 without visual feedback. You can if you're sober, you can close your eyes and you can bring the tip of your finger and the tip of your nose into contact with one another. Because your brain is being given information about, even though you're not looking at it, about where the tip of your finger is. You can take the tip of your finger and you can touch the tip of your ear or the top of your head or your other shoulder, right? Without looking at it. So the kinesthetic system is giving you feedback about where your body is in space. And this is the sensory system that determines, in no small part, to what degree are you a good athlete? To what degree are you clumsy or not? This is one of the sensory systems that we see impacted not infrequently in cases of autism. The kinesthetic system, the proprioceptive sense in somebody on the autistic spectrum is often impaired, and you get the impression accurately that often the person with autism doesn't have the sense of kind of like where they end and the rest of their environment begins. This causes problems for kids with autism. The classic event being kid with autism walking down the hallway, grooving along. Some other kid is coming toward him from the other direction, and generally in that situation, what you do is you're walking along, and another person is let's see if I can get my hand in the frame here. Another person is coming along towards you, and you have an awareness of of what space your body is occupying, and as that person comes towards you, this is the case for everybody, but particularly if you're an adolescent male. Males in general, especially adolescent males, are very, very sensitive to any cues about aggression. If you do not want to communicate aggression, one of the things you do when somebody's walking along is you each turn your body so that your bodies don't touch as you walk by each other, right? If you don't do this, somebody's walking along, you're walking along, they give a little bit, they turn their shoulder, but you don't, and you bump them, and that just animalistically, very primitively, is communication of an aggressive act. Well, in order for you to be able to make this move to let the other person by, you have to kind of have a sense of where the tip of your shoulder is. You have to have accurate proprioception for that part of your body. And for somebody on the autistic spectrum, they often don't get good feedback, don't get good accurate feedback about that. So, and there's other complicating factors in here too. One of the complicating factors is they're not intuitively picking up on social expectations for things like what might be experienced as aggressive by other adolescent males. But even disregarding that, say they get a little bit of that, or they've seen it and they're mimicking the kind of giving way so you don't bump shoulders. They do this, but they don't have a good sense of where it is, and they bump the other guy. And the other guy's reaction is, what are you trying to start something? And you have a little scuffle. This kind of thing. And the good news is you can learn this with some practice. This is part of what you're doing if you are picking up a new instrument, or you're learning how to shoot a free throw or something like that. You're developing certain pathways in the proprioceptive system. You're pruning. You're eliminating unnecessary connections and strengthening the appropriate connections so that you're getting good, efficient feedback about where your body is in space, where your joints are. The other component of the proprioceptive system is strength of movement. How forcefully are you contracting the muscles to move a limb, right? That having a sense of that makes all the difference. It makes a difference between picking up a cup, say it was a solo cup, picking up a solo cup and picking it up and putting it to your mouth, or picking it up and crushing it, right? You have to be getting good, accurate feedback from the muscles in the joints in your hands and your arm and your shoulder and everything, so that movement comes off without a hitch. I indicate here on the slide that it's a fairly new area of study. New in this case is relative. It really kind of came into its own in the 70s and 80s, and I was speaking a few minutes ago about people with autism. It is a particularly pertinent, occupational therapists, particularly pertinent for people with autism in terms of helping to rehabilitate, or to helping to develop some of these skill sets. So the kinesthetic system giving you information, giving you proprioceptive information about body position and strength of movement. The other of these, the other part of the somatosensory system, the other tactile-based system, is, I referred to it a few minutes ago, is the vestibular, the balance system. And the balance system detects acceleration and changes in movement, and it is a function of part of the inner ear, what we call the semicircular canals in the inner ear. And when we start talking about, it's going to be maybe a little confusing, but when we get to talking about auditory experience, when we actually break down the inner ear, I'll show you what I'm talking about in terms of these structures in the inner ear that look like arches on the inner ear, and they're actually tubes that are arches that are filled with fluid, and inside these arches are the ends of the neurons that give you information about balance. And the ends of these neurons are, they're quite literally cilia, hair, cilia meaning hairs. There are cilia that are in these tubes. They stick out into the interior of these tubes, which are filled with fluid. And when you turn your head, when you tilt your head forward, backward, the fluid in these tubes moves, and it moves the hair-like ends of these neurons, which causes the neurons to fire and gives you information about where in space that your head is. And that's a large component of the experience of balance. Now, keep in mind, you're also getting feedback from the kinesthetic system, the proprioception. Where are my arms and my legs and my hips and my torso and everything in space also. So that is a component of your experience of balance. And like I said, I'll talk to you a little bit more about where this is and what this looks like, the actual structures involved, where they are and what they look like when we actually get to talking about the inner ear, when we talk about auditory sensory experience. But first, we will start off talking about the translation of electromagnetic radiation into electrochemical energy. So the translation of light energy into action potentials, into the electrochemical messages that your nervous system uses. The neurons that are responsible for this translation of light energy into action potentials are the photoreceptors that make up the back of the inside of your eyeball that comprise the retina. And this light energy is most helpfully thought of as coming in waves, with wavelength corresponding to color. The more compressed the waves, the shorter the wavelength, falling at the blue end of the spectrum, of the color spectrum. If you think of a rainbow, blue transitioning into green, yellow, orange, and red, right? And then red, that corresponding to longer wavelength, right? So short wavelength light comes into the eye and is experienced as blue, or toward the blue end of the spectrum, and the longer wavelengths come into the eye and are experienced as toward the red end of the spectrum. How relatively bright or dull the color is corresponds to the height of the wave, the amplitude of the wave, and how pure, how vivid the color is, how vividly red it is a matter of how pure the wavelengths are. Are you getting all of one kind of wavelength coming into the eye? Okay, so before we talk about the photoreceptors that actually transform these waves of light energy into action potential so your brain can do something with it. Let's talk about the basic structure of the eye. I'm going to show you on this diagram the vocabulary that I want you to be familiar with and the vocabulary that I'm not so concerned about. Let's do this. I am not concerned about you knowing the names of the muscles, the ciliary body, the suspensory ligaments, the posterior chamber. I hope you're thinking about how nice I am. The lacrimal caruncle, the other muscles, choroid. I'm not suggesting that these things are not important. What I am suggesting is that I know you have a limited amount of space in your head for storing stuff, and I want you to focus on some of the things, these more basic structures that I would say are more important for our purposes. I want you to be familiar with the scientific term, the medical term for the white part of your eye, which is the sclera. The sclera is actually a remarkably tough outer layer of the eye. If you've ever been poked in the eye, probably the first thing that occurs to you is it's extraordinarily painful. You have a lot of sensory neurons and there's a tremendous pain sensitivity for the eyes, predominantly because they are so profoundly important. We have a remarkably large portion of the brain devoted to processing visual information, which is an indication of just how important the eyes are to our survival. In case you needed some sort of evidence. So because of how important the eye is to our survival, and because of how much brain is devoted to associated processing, the body, the brain, evolution protects the eye by making it very pain sensitive. That being said, if you have ever been poked in the eye, one of the things that you may have noticed is actually how durable that part of the eye is. Yes, you can scratch it, and yes, it will bleed. But we tend to think of our eyes as very, very fragile, a very, very fragile part of the body. But in fact, the sclera and the cornea, which is essentially the continuation of the sclera, as it moves across the front of the eye, it becomes clear. And it's through the cornea, through the clear part of the outer covering of the eye, that you can see the pupil, which is the black center of your eye, that is the actual opening through which light passes to the interior of your eyeball, and the iris, which is the muscle that controls the size of that opening. So the cornea, the sclera, both very tough, both obviously very pain sensitive to protect something that is so very important. The iris, the muscle that contracts and relaxes to control the amount of light entering the eye through the pupil, which is the opening itself. Behind the pupil is suspended, is a round lens that is surrounded, just like the iris on all sides, by muscles. So you have the iris, which is a muscle that expands and contracts to control the amount of light entering the eye, and then directly behind the pupil in the iris is a lens, which is a malleable, clear lens that is surrounded on all sides. It's attached by muscle to the eye wall itself, and those muscles contract and relax to change the shape of the lens. And what that does is as it changes shape, it helps to focus light at a particular spot on the back of the eyeball, depending on whether it's very bright out, in the case of the red lines that you saw there, in which case when there's a lot of light available, the iris makes the pupil smaller, which narrows, which reduces the amount of light entering the interior of the eye, and the lens changes shape to focus the light into the center of the back of the eyeball, into what we call the fovea. And we're going to talk about that in just a second. When there is less light, the iris expands, it relaxes, and the muscle opens up, the pupil gets larger, and the lens, again, changes shape, and we'll use green here. And what it does is it allows light to spread over the back of the eye a little more broadly, like this, like you can see now depicted with the green lines there. And basically what the lens is doing is focusing light where it will be most useful. In the retina, the very center of the back of the eyeball, the fovea, what you see described there as the central depression, is a depression because when they say it's a depression, it looks like a little hollowed out section at the back of your eyeball. It looks like a pit, and the reason it's a pit is because the neurons, the photoreceptors at that part of the eye, are actually shorter. They're cones, and those photoreceptors are very light sensitive, very detail sensitive and color sensitive, but not particularly light sensitive. They require a lot of light energy in order to work efficiently. But they're very, very good at detecting detail, and they're very good at detecting color. When light levels are lower, and the light level that's available is not enough to stimulate those cones, those photoreceptors that are sensitive to detail or productive of detail and color, what happens is the iris relaxes, the pupil opens wider, and the lens distributes the light that is available, what little light is available, across more photoreceptors at the back of the eyeball, to include the rods. And if this isn't making any sense to you, wait, and we'll talk about this more on the next slide. Just in terms of anatomy, we've already talked about the sclera, the iris, the pupil. I think I made mention of the cornea, which is basically, it's essentially the same kind of material the sclera is. It's very tough, durable coating, but it's clear, whereas the sclera is white. When the light enters through the cornea, it passes through what we call the anterior chamber, and the anterior chamber and the vitreous body, which is the interior of the eye, are both filled with fluid. When in the anterior chamber, we call the fluid the aqueous humor. On the interior of the eye and the vitreous body, we call it the vitreous humor. But it's clear fluid, and the clear fluid is there partly to deliver nutrients and remove waste and so forth, but also to actually provide some structure to provide some support for the structure of the eye. Filling it with fluid and having some degree of pressure on the inside of the eye makes the eye more structurally sound. It makes it less susceptible to damage. So anterior chamber filled with aqueous humor, vitreous body filled with vitreous humor. The retina, which is depicted there in kind of an orangish yellow, that is the actual layer of neurons on the back of the eyeball. The fovea and the macula, which we'll talk about here more in just a second, and then the optic nerve and the optic disc. So what the optic disc is, if you can imagine this, so the back of the eyeball is essentially all of these photoreceptors that are compressed in very densely, and they make the entire back of the eyeball is all of these photoreceptors kind of lined up or squashed in side by side. And each of those receptors has an axon. And it becomes a little more complicated, and we're going to talk about that complication here in just a second. But essentially, axons from these receptors have to be bundled together and exit the back of the eye, because that's the bundle. Those axons are the cable, so to speak, along which the action potential message will be sent to the back of the brain, to the visual cortex. And what the optic disc, the blind spot, is the point at the back of the eye, which, if you're looking directly at the eye, it's the center of the back of the eye. If you look in through the pupil, is that fovea, is that central depression. And if you look down just a little bit and a little bit in towards your nose, there's the optic disc, the blind spot. And the blind spot is where all of the bundles, all of the axons from these receptors are all bundled together and exit the back of the eye. The reason why it's a blind spot is because where it's all bundled, there are no receptors. What's there are the axons, right? So there is a blind spot, and it's actually fairly fascinating that while there is nothing there, there's no receptors there to process visual information, your brain actually accounts for the blind spot and fills that blind spot in with information gathered from photoreceptors surrounding the blind spot. So generally speaking, you're never aware that it's even there. And then, of course, the bundle of these neurons, these axons from the photoreceptors comprise the optic nerve, and of course the vessels, the blood vessels and such that supply the eye with nutrients, all bundled out the back of the eyeball. I'm trying to think if there's anything else here I want to make sure you are aware of. There is not, so I will go to the next slide. Okay, so you can see on this slide a diagram on the left there, fairly similar to the previous diagram. I have this slide in here because I wanted to show you a couple of things. First one is what I was talking about when I was talking about the blind spot in the previous diagram. This is kind of blown up so you can see the axons in yellow there bundling together from the rest of the retina, and of course the blood supply from the retina bundling together to form the optic nerve, which exits the back of the eye. And you can see where the blind spot is depicted with the bracket there. You can see where there are no photoreceptors. The photoreceptors in the human eye are what we call rods and cones, and you can see those depicted in red for the rods and cones, or in green for the cones. So that's the first thing I wanted you to see, was that little better depiction of exactly why there is a blind spot, because of course there's no receptors available there. The other thing that I wanted to revisit just briefly is the fovea, which sits at the center of the macula. You can see it depicted here on the left of the diagram, the fovea centralis. And essentially what the fovea is an area in the retina where you find exclusively cones, which are the photoreceptors. I think I may have mentioned in the last slide there. Cones are the photoreceptors that are responsible for giving us the experience of color, vision, and for good detail. They're kind of the HD photoreceptors. So cones, high definition color vision. Rods function much better in lower light levels, but are not as high definition, I guess you could say. They don't give us the acuity of the information that the cones do, and they don't produce color vision. So this is the reason why when you turn out the light in a room, or you're outside at night, you don't see vivid color. Now the cones are still somewhat active in low light conditions, even in moonlight, say. But they don't work as effectively, so you get a dramatically muted experience of color, because predominantly the information that is being delivered to the visual cortex is coming from the rods. To back up for just a second, what I started to say before is the fovea, the center point in the back of the eye on the same axis as the pupil and the lens, is where the lens is focusing the light when there is plentiful light. And what's happening there, the fovea is the area in the retina where it is exclusively cone photoreceptors, meaning the fovea is the point in the retina where you have the photoreceptors that are responsible for seeing vivid detail in color. So when there's enough light, the lens, the cornea, and the iris and the lens operate to focus the light onto the fovea, where all of these high definition color sensing cone photoreceptors are. As the light levels diminish and the iris contracts and the pupil opens and the lens shape changes so that it distributes the light more broadly across the back of the retina, this is generally what you would expect with lower light levels. The light is then focused onto more and more rods. It's of course still hitting the cones, but it minimally, their lower light levels don't do a lot to stimulate the cones. They don't function very well when there's low light levels. So as you move away from the fovea, where all the cones are concentrated, the area immediately around the fovea, so you have the fovea, the area immediately around it, the macula, is essentially kind of where you start to see a transition from cones to rods. So the macula is, you still have cones present, but you're beginning to have they're beginning to be interspersed with some rods. And then as you move out from the macula, the back of the retina is predominantly predominantly rod photoreceptors. So the other thing I wanted to make sure I mentioned to you is the fact that we have what is sometimes called an inverted retina. And what I'm referring to here, if you look into the place to the box there where it's kind of the blown up depiction of the back of the eyeball, you can see the yellow arrows that are to depict the light rays being focused from the lens coming in through the pupil and being focused by the lens on the back of the eye. And what you'll see there is the rods and the cones, the tips of the rods and the cones that are the actual kind of, the photoreceptor itself is actually facing what appears to be the wrong direction. They're pointed toward the back of the eye. And this, what initially appears to be a pretty bad design, a pretty inefficient design, is, from what I have seen, the explanation is that this is in part to put the rods and the cones behind a fairly substantial blood supply. And the reason that is happening is because the blood supply actually serves to reduce the temperature on the back of the retina when light is being focused there, because if you don't control the temperature, you very quickly damage the cones and the rods also, of course, but the cones specifically are damaged by variations in temperature. So they're protected essentially by these layers. The other thing I want to make sure that you see, in addition to that idea of the retina being inverted, is that the rods and cones are so dramatically numerous in the retina. There's so many of them that if you tried to bundle all of the axons individually from the rods and the cones and run them out the back of the eyeball, the whole back of the eyeball would be axons. There wouldn't be any place for photoreceptors because there's so many of them. And the way this problem is handled in the eye is that you can see here that the information from multiple photoreceptors, say from multiple cones, is condensed from by what they call bipolar cells and then to ganglionic cells. So what you have is you might have 4 or 5 cones, making this up numbers wise, 4 or 5 cones that terminate on a bipolar cell. So all they're sending all of their information to one bipolar cell, and then there might have 4 or 5 different bipolar cells, each receiving information from 4 or 5 cones, say. And then these 4 or 5 bipolar cells, which are responsible altogether for 25 cones, again, making up the numbers, all terminate on one ganglionic cell, so that information coming from, say, 25 cones ends up being delivered out of the back of the retina along one axon. So it's simply a way of making the number of axons manageable so the eye can actually function, so you can actually have enough room in the back of the eye for photoreceptors. So bipolar cells, the thing I want you to understand here is that bipolar cells and ganglionic cells serve in part to condense the number of axons leaving the back of the eye. And I think that is everything that I wanted you to be aware of on that. Yes. So you will see on this slide the information about rods, cones, bipolar, and ganglion cells, so that you don't have to spend much time writing all this down. The definitions and descriptions are there for you. I also wanted to include this, which is an actual photograph taken through the pupil, so that you can see. It's a little off center because the macula in an actual eye would be dead center in the middle of the eye. Remember, the macula and the fovea are the areas of the eye where you have the rods in the periphery of the retina gradually being interspersed with more and more cones until you get into the fovea, which is kind of the dip in that depression in the back of the retina, until you get to the fovea where the photoreceptors are exclusively cones. And then you can see the point at which all of the veins and arteries come together is the point where the optic nerve is leaving the back of the eye, and that is that blind spot at the back of the eye, because there are no photoreceptors there, because it is all blood vessels and axons leaving the back of the eye. Okay, and I've added this one in, this slide in, because I wanted to give you a picture of what nearsightedness, myopia, and farsightedness, hypermetropia, look like in terms of the structure of the eye. One of the things that you'll notice there is that in somebody who is myopic, one of the problems is the eye is often elongated. It's shaped a little more like a football, whereas in farsightedness, the actual axis of the eyeball is shorter. And so what's happening is that the lens, when the lens is functioning properly, is actually focusing the light so that it is not perfectly focused into the macula and fovea at the back of the eye. In myopia, it's focused too early, and in farsightedness, it's focused essentially, hypothetically, where it would come to a focus beyond the back of the eye. And I just wanted to have you have that general picture of what the details of what nearsightedness and farsightedness actually look like. And before we move on to talking about the structure of the ear and auditory processing, I want to just make a brief mention of the area of the brain where the information that is gathered by the eye, the sensation, the data supplied by the eye, by the photoreceptors in the retina, and sent along the optic nerve, which eventually terminates on the occipital lobe, which we talk about as the primary visual cortex, the occipital lobe, of course, being in the back of your head. This area of the cortex is where the visual information is assembled into, the sensory information is assembled into the actual experience of vision. And the process of visual perception is immensely complicated. There is some discussion of it in your book, and I would encourage you to look over that closely. I do want to mention here that one of the primary means by which the sensory visual data is processed into visual perception is by what we call feature detectors. So this would be these would be cells that are adjacent to one another in the visual cortex that are stimulated by sensory data of, say, a vertical line or a horizontal line, or each in its own cell lines at varying degrees between 0 and 90, say, and then beyond 90 toward 180. So there are individual nerve cells that detect lines, edges, say, at specific angles. There are feature detectors that are tuned to particular colors. These kinds of things, and what happens is that the sensory information is delivered from the eye to the back of the brain, the back of the cortex, the occipital lobe, primary visual cortex, and say information. Say the image that you're looking at is a drawing of a three-dimensional cube that is blue in color. The visual information is delivered to the occipital lobe, and feature detectors that correspond to the horizontal and vertical lines that represent the front and the back of the cube, and of course the diagonal lines that represent, that give it the three-dimensional aspect. You will get these individual cells that correspond to diagonal, vertical, and horizontal lines, and feature detector cells that correspond to the color blue, that particular shade of blue, all light up simultaneously. That information is delivered to other parts of the brain that then take those feature detections and put them together so that you have the seemingly instantaneous recognition of, oh, blue cube. So remember this concept of feature detectors. We see this also in auditory processing. The other thing I wanted to make sure I mentioned to you, because it's a fairly well-known, fairly commonly talked about visual processing phenomenon, is this thing that we call inattentional blindness. And this is essentially just the recognition that a way of distinguishing perception from sensation is that you can have sensory data delivered to the visual cortex. So in other words, the information is coming into your eye, it's encoded, or it's translated into action potentials by the rod and cone photoreceptors. It's delivered along the optic nerve back to the visual cortex. Feature detectors light up corresponding to this information that's being gathered by the eye, and then it is processed in terms of creating the pictures, creating the perception, creating the images that have meaning to you. Well, in the course of this, one of the things that we see is that often, and not often, inevitably, the information that is being processed is fine-tuned, it's narrowed, and some of the data that's coming into the back of the eye is almost, I guess you could think of it as being disregarded as unimportant. So if you're watching the little video of me as I'm talking to you in the lower, would that be the lower right-hand corner of your screen, I would assume, maybe at the outset you attended to certain things in the background of the room that I'm sitting in, the books on the shelves and the globe in the upper, on the top of the shelf there, maybe even my beautiful fly rods above the bookshelves. But maybe initially when the picture is novel, you might have noticed those things, but you become completely captivated by the lecture. You get focused on my face, and me talking, the movements I'm making, and after a while, you begin to divert attention away from what's in the background, and you don't see it anymore until somebody calls your attention to it, and then of course you shift the attention and it becomes visible again. It's not that you don't. It's not that you aren't sensing it. You of course are sensing it. I think I may have talked to you earlier in this semester about a famous experiment where I think it's been done a couple different times. Once where they had teams of people throwing basketballs back and forth, and the subjects in the experiment were asked to count the number of ball throws. And then in another one, I want to say there was a play acted altercation, a tussle. And in each of these, in the course of either counting the ball throws or this little tussle, they had a person in a gorilla suit walk across the screen. And what they found was, I want to say it was more than half of the people did not see the gorilla. Their attention, their processing was focused on a particular part of the visual picture, and while certainly they sensed, in other words, they had the sensory data detected by the photoreceptors in the retina, delivered along the optic nerve to the primary visual cortex, and detected in the primary visual cortex, but then not assembled into the experience of vision. So when you ask the person, what did you see? And they report, well, I saw them throw the ball back and forth 27 times. And you ask them, well, did you see the gorilla? And they say no. In fact, they'll often not believe you until you show them a video of it, and then they're alarmed at the fact that they didn't see the gorilla. This is not just visual. We do this with auditory information and other information all the time. And it's necessary. We need this. If you're incapable of doing this, essentially what develops is ADHD, right? If you can't screen out visual information, auditory information that's not pertinent to what you're doing, and so you're paying attention to everything all the time, what ends up happening is you become highly distracted. You're not able to, distracted is basically you're not focusing on the pertinent information. You're focusing on things that are not central, right? So this unintentional blindness is not actually a weakness, it's a strength. It can be a weakness if you if you misperceive something as unimportant, and in fact it is important, but this inattentional blindness and cognitive processes, perceptual processes related to it are necessary for us to be able to function in a world where the information, the sensory information is so immensely complex. Okay, so let's shift gears a little bit. Let's talk some about hearing and about the anatomy of the ear. The auditory processing takes place in the temporal lobe, which is located about where you would expect it to be. The receptors, we talked about the retina being comprised of photoreceptors, neurons that are sensitive to light energy. The neurons in the auditory system are mechanoreceptors that we call cilia, which means hair, and we call them that because these mechanoreceptors look like little hairs. And they are stimulated mechanically. They're stimulated by actually being moved. And we'll talk about how that takes place here in just a minute. Okay, so as with vision, we talk about the stimulation of the auditory nerve in terms of waves, in this case sound waves, vibrations in the molecules in the air. And we break this sound quality down according to pitch, which corresponds to wavelength, how close together or far apart the waves are. Amplitude, how big the waves are, how tall the waves are, and that corresponds to how loud or soft a particular sound is. And then purity, which we talk about as timbre, and that's essentially the tonal quality of the sound. And you can kind of think about it like this, like you can have the same note played on different instruments, but that note is it can be played at the same volume, same pitch, but it's going to sound different coming from different instruments, and that would be the timbre, the purity of the note. I'm just listing here for you the terms for the parts of the external, middle, and inner ear, but rather than go through them on this written slide, I'm going to jump to the picture of the ear. Okay, so I'm going to do with the ear diagram the same thing that I did with the picture of the eye. We're going to go through the terms that I want you to be familiar with and talk about the basic function of the ear. I will do this. And I will omit the things that I'm not real concerned about you knowing. I don't need you memorizing the carotid artery, the styloid bone, and that's the temporal bone. But that's about it. Everything else, pretty much everything else on here I want you to be familiar with. Okay, so I want you to be familiar with the actual scientific term for the external ear, which is the pinna. I want you, and essentially the purpose of the pinna is as a funnel. It's designed to funnel the sound into the auditory canal. It's also designed to help for directional detection also. But the pinna funnels the sound into the auditory canal. The sound waves, then compress the tympanic membrane, your eardrum, and the movement of your tympanic membrane, your eardrum, with the sound waves moves the three tiny bones of the middle ear, the hammer, anvil, and stirrup. I do want you to know the technical terms for the hammer, anvil, and stirrup, just because that they're fairly commonly used. But the vibration of the eardrum moves these bones. These bones terminating in the stapes, the stirrup, sits on what they call the oval window, which they don't have listed here because underneath the stirrup. But the oval window is a membrane-covered opening on the inner ear. In the inner ear, that thing that looks like a snail with three arches coming out of it, is actually hollow and fluid-filled. And so what's happening is as these bones of the middle ear are moved by the vibrations in your tympanic membrane, your eardrum, the stirrup moves, vibrates against that oval window, and when it does that, it causes waves in the fluid that fills the inner ear. You'll see there the round window, which is just a little bit below and to the right of where the stapes sits on the inner ear. The round window is another membrane-covered opening in the inner ear, which is hard. The inner ear itself is an unyielding shell. Well, the round window is another membrane-covered opening, and essentially what that is it can bulge out so that when the stapes presses against the oval window to make waves in the inner ear, the round window bulges out so that the waves can happen, right? If you didn't have a round window, it would be essentially impossible to make the waves, or not impossible, but the sound would have to be much, much more forceful to make any waves at all. So the round window there is essentially to kind of allow the pressure to fluctuate so that waves can be created within the inner ear. So the cochlea, the part of the inner ear that looks like a snail, is actually where the sensation of hearing is produced. And then the semicircular canals we talked about earlier in the lecture, and that is that they're where you get your vestibular sense, your balance sense. They're also fluid filled. They also function via mechanoreceptors, cilia, just like in the cochlea. And these cilia, these hairs, project out into the fluid that fills the semicircular canals, and then as you tilt your head forward, backward, side to side, the fluid moves along these semicircular canals, moves the cilia, the little hairs, and that stimulation is sent along the vestibular nerve to send information about balance sensation to other parts of the brain. Similarly, in the cochlea, we're going to talk about the structure of that here in just a second, but the cochlea also is filled with these nerve endings, these hair-like nerve endings that we call cilia, and it is the pressing of the stirrup up against the oval window that makes waves in the fluid. As the waves in the fluid move, they move the little hair-like, the cilia, nerve endings, and that produces action potentials that are sent along the cochlear nerve, or the auditory nerve, to be processed in the auditory cortex. The eustachian tube is the opening into the mouth and throat. It's the part of of your head that's essentially leading to you getting that clogged up feeling when you get pressure changes, like if you're going in an airplane or you're going up a mountain or something like that. And that's the reason why yawning, making that pop in your ears, what you're doing is you're opening the eustachian tube so that the pressure in the middle ear where the bones are becomes the same as it is on the other side of the tympanic membrane, the eardrum. Make sure I hit all the vocabulary there. It looks like I did. All right. So here I just wanted to give you another picture, so to speak, of the cochlea, so you have an idea of kind of maybe you're able to picture a little better the actual mechanism by which the sound is being mechanically produced. Okay, so you see the stirrup there, the stapes, and it's pressed up against the oval window, which is the opening in the membrane of the inner ear. And as sound waves press on the tympanic membrane, it moves the three bones ending in the stapes. The stapes moves against the oval window. It creates waves that move along the inside of the cochlea, and you can see they move, I guess you could say, down along the snail shell. And what they show you there in the upper part of the picture is the cochlea, what it would look like if it were unwound and open. The waves in the fluid move along the what we call the vestibular canal, and which eventually terminates at the round window where pressure is relieved, so you actually do get waves. I am not going to ask you to know the terms perilymph, apex, which is just the smallest point of the cochlea, basilar membrane. I do want you to know what the tympanic canal is. That's just the tube that is on the inside of the vestibular and the tympanic canals are just the inside of the cochlea. Stapes, oval window, round window. Don't need to worry about knowing what the base is. And then I do want you to be aware, you can see the kind of twisting here of the axons of the sensory neurons. And the reason it looks twisted is because when you roll, they're showing you the cochlea unrolled, so to speak, but as you roll it back up, those axons bundle into a fiber, the actual auditory, the auditory nerve. Okay, we'll shift now to talking about smell, sense of smell, olfaction, and taste, gustation. We're going to talk about them adjacent to one another because they're related to one another, but they are both mediated by chemoreceptor neurons, neurons that are stimulated by chemical shapes. In the case of the olfactory chemoreceptors, which are cilia, again, hair-like extensions into the upper part of the nose, which I'll show you a picture of that in just a minute. There are about 350 different kinds, distinct types of these receptors. And what I mean by that is that each of these 350 different kinds of olfactory chemoreceptors are designed to pick up to be stimulated by one particular kind of chemical. And all of the kind of the rich nuance in sense of smell is essentially various combinations of these approximately 350 different kinds of specific chemical receptors. The other thing to be aware of with the sense of smell, there are two other things to be aware of. The first one is disgusting, and that is that the way that these chemoreceptors that provide you with your sense of smell are stimulated is by actually having physical contact with the chemical, kind of like what we talked about when we talked about neurotransmitters communicating a message between neurons, that the neurotransmitter kind of plugs in to a receptor that is designed for that particular neurotransmitter, that particular chemical. The shapes fit. Well, a similar thing is going on here with the chemoreceptors that they have. They are a receptor designed to fit a particular kind of chemical. Well, in order for the chemical to stimulate that receptor, it has to be inhaled. The reason why I say this is disgusting is because the implication of this is that when you smell, say somebody passes gas and you smell their fart, well, guess what? This is the disgusting part. What you're actually smelling are the little bits of butt matter, the actual vaporized chemicals that were in that person's rear end. They pass gas, it distributes out into the area around them. When you smell it in, what you are actually smelling in are the chemicals that came directly from that person. Another way of saying this is you're walking outside and you walk by a pile of dog poop and oh man, that smells terrible. Well, the only way you smell it, one of the things you'll notice is that if it's very, very cold out, so cold that poop on the ground is frozen, you're not going to smell it. The way that you smell it is that it has to be volatile enough that some of the chemicals that comprise whatever that object is have dissipated into the atmosphere around it. And so when you smell that poop, you're actually, the way you smell it is that actual molecules from the poop have been vaporized, have gone into your nose, and are now touching the olfactory cilia, the chemoreceptors that allow you to smell that nasty smell. So you are quite literally smelling ass matter. So that's the one thing. That was the gross thing. The other thing to keep in mind about the sense of smell is that it is the one sensory system that's not rooted through the thalamus. And one reason why we think that this might be the case, or how this arose, is that it speaks to just how ancient the sense of smell is, that there's some evidence that it actually probably was present in brains before other senses were being routed through the thalamus. But the other thing to keep in mind is that because it's not routed through the thalamus, and because it goes directly from the olfactory bulb, which I'm going to show you in just a second. Actually, I will jump to that right now. So this is the image of what we're talking about here. You see the nasal cavity there with the fibers of the olfactory nerve permeating through and extending from the cribriform plate, the bone that comprises the top of your nasal cavity. So you breathe in whatever chemicals make it past the hairs and the tissue and the moisture on the inside of your nose and actually make contact with the chemoreceptors that are your olfactory nerves, plug into the particular slots that are designed for those chemicals, cause an action potential to initiate up through the cribriform plate and then into the olfactory bulb. That stimulates other neurons in the olfactory bulb, and those other neurons have axons connecting them with directly to the olfactory cortex, which we're going to show you in just a second, and to the limbic system directly. And this is one of the reasons why, remember, the limbic system is kind of the emotional brain. This is one of the reasons why a sense of smell has such kind of visceral, primitive, emotional content, not infrequently, right? A smell can bring back a very distinct and sometimes a very distinct memory, and sometimes a memory that's hard to put into words because it isn't necessarily being concurrently processed by the parts of your brain that are responsible for language. So you'd have a smell. It's routed directly to the part of your brain that actually organizes the sense of smell itself, the olfactory cortex, and to the limbic system where it's being you're having an emotional response, perhaps, depending on what the smell is, to the smell prior to you consciously processing the implications of that smell. And this is partly to keep you safe, right? You know, part of the sense of smell is a very, very, very primitive smell, and part of the purpose of it, hypothetically, in an evolutionary sense, is to protect you. You know, you're very sensitive, and our ancestors certainly were very sensitive to the smells of certain animals. Infants are born like being very, very, very sensitive. Apparently, essentially from the moment of birth, being programmed to have a sense of smell where they can detect the scent of their mother's milk, even from a fairly significant distance. So it's a very primal survival mechanism. I have this slide in here just to give you a little more detailed picture of the transition from the extension of the olfactory receptor cells, the chemoreceptors sticking out in the nasal passage, the nasal cavity, and then of course distributing up through the cribriform plate, the bone, and into the olfactory bulb, where they connect with other neurons whose axons extend to the limbic system and to the olfactory cortex, which you see there is essentially directly above where the olfactory bulb is. I'm not going to say much more about smell, other than, again, to make the note of the fact that it is very closely related to taste, and it is responsible for a substantial percentage of your experience of taste, right? You know, if you hold your nose and you put something in your mouth, you hold your nose and you take a bite of a lime or a lemon, and you'll be able to tell that it has that, I guess it's a sour taste, but you won't be able to distinguish necessarily, is it a lime or is it a lemon? Once you unplug your nose and you allow yourself not to just taste it, but also to smell it, you can make that distinction. And part of the reason for that is, remember how we talked about there's 350 different chemoreceptors that are part of smell? Well, there's only basically 5 different receptors for taste. You got sweet, sour, bitter, salty, and then more recently they've identified something they call umami, which is essentially kind of the sense of savory or meaty flavor. MSG, monosodium glutamate, is what you're tasting there is umami. So at any rate, gustation, taste, is, as I said before, also mediated by chemoreceptors. The chemoreceptors, these are not your taste buds. They are clustered around the bumps that you think of as your taste buds. taste buds. And essentially anything you taste are chemicals that have been dissolved in your saliva. If you don't get some solution as a result of dissolving in your saliva, you don't have a taste, right? So that's why things that can't dissolve at all, you're not going to taste. And similar to the sense of smell, it's routed to the frontal cortex, to the gustatory cortex. Hard to say. I'm not going to say much more about this. If you get into evolutionary psychology, perhaps into developmental psychology, and certainly into biological psychology, you'll talk a lot more about that. More on the smell end of things than the olfactory end of things, than the gustatory end of things, the taste end of things. And the last thing that I wanted to make sure I went over for you is the tactile system, the sense of touch. Now, I'm making a little bit of a distinction between the tactile system and the somatosensory system. Remember, the somatosensory system would include the proprioception and balance, right? Vestibular and proprioceptive experiences. But in this case, when we talk about the tactile system, we're talking about specifically your sense of touch, your sense of feedback from the mechanoreceptors and thermoreceptors in your skin, the pressure sensors and the pain sensors and the temperature sensors. They're routed to the somatosensory cortex, which I think I will show you in the next slide, I believe. The somatosensory cortex is on the very edge of the parietal lobe, right next to the motor cortex. They're adjacent to one another because they have a significant relationship. Essentially what's happening, part of what's happening is that motor movement is related so very, very closely to tactile information. So you touch something, you get a pinprick, you touch something that's hot, you touch the eye of a stove and it's hot, and that information, the heat information from the thermoreceptors goes up, processed by the somatosensory cortex in the parietal lobe, and which is immediately adjacent to the motor cortex. Message is sent to the motor cortex, and very, very quickly the message goes down through the brain, through the spinal cord, down along the arm to the hand to get you to send the message to remove your hand from that eye of the stove before you burn yourself irreparably. When we talk about the somatosensory cortex and we talk about the tactile information processed by the somatosensory cortex, one of the things that is often mentioned is this idea of a homunculus, which is the depiction of the organization of the sensory processing along the somatosensory cortex as if it were a person. And it's probably easier to show you this than to try to explain it. So I'm going to advance to the next slide. So there you'll see in blue the somatosensory cortex, and what this slide is depicting is if you were to take a cross section that is that somatosensory cortex and look at it, what you would find is that information corresponding to different parts of the body is processed in very predictable parts of the brain, and the amount of information that is received from these various parts of your body corresponds to the relative area of the brain devoted to it. So the homunculus is a picture of a little person where the size of, in this case, like if you look at this picture, the hands, the tongue, the mouth are depicted as quite large. There's a lot of sensory information coming in from those parts of your body and being processed in the somatosensory cortex. If you think about what your hands and fingers are responsible for, if you think about how fine control you must have over your lips and tongue in order to produce intelligible speech, it would make sense that there's a significant amount of input coming, sensory input coming from these parts of your body. I'm not going to say much more about this. I told you I would try to keep this to less than what our typical class period would be. So I am going to stop here. I'll mention to you, I'll probably also make a note of this in an email to you, that I'm going to throw in some crash course, some links to crash course videos for this same material, hopefully to make some of this a little easier for you to understand, since we're not doing this lecture in person. So I will stop here and try to get this uploaded to YouTube, which will take a while.